coming up from the subsurface. It can also be an indicator, perhaps, of a living system on Mars. And of course, there's been an array of instruments sent up over the years, including having our eyes ahead on a Mars sample return in the coming decades. And so, targeting subsurface environments on Mars, considering whether they're habitable, and considering what to look for are all important considerations. And particularly, this is the European Space Agency's Rosalind Franklin rover. This is specifically targeting accessing subsurface environments of Mars, obtaining at least shallow cores from in the Martian subsurface environment. Another really exciting target are these ocean worlds of the outer solar system, Europa, Enceladus, Titan, and others. And these are places where liquid water exists, but it's underneath a very thick ice cap. And so here again, to access the habitable environments where the liquid water exists, you need to look at the surface or things you can actually detect. We don't yet have a means to drill through 100 kilometers of ice on one of these ocean worlds. And so we can look at the cracks in the surface on Europa. We can look at plumes coming out of Enceladus. We can look at hydrocarbon lakes on the surface of Titan and look for clues of subsurface life spilling out into these places. And just to quickly move through them, Europa has been a very attractive target because it appears to have both the liquid water, subsurface ocean, and possible volcanic activity, which, as we've seen on Earth, supports very distinct and vibrant life forms already. And importantly, just last Friday, the JUICE mission was launched by the ESA, shown in the right here, with the sloth in the foreground, which is going to arrive in the outer solar system by 2031. And this is looking specifically for signals and learning more about the habitability of Europa and some of these other ocean worlds. Enceladus is exciting. This is a moon of Saturn because it has these plumes going out into space, and so it makes it a little bit easier for orbiters and spacecraft flying through this to potentially characterize the chemistry. It's ejecting material from the oceans into space. And so this is another exciting target. And so they've developed possible missions there that are still in the works, but look into the future of having orbiters and landers concepts. And then Titan's really exciting because it's a large moon and it has an atmosphere. And so there's been a mission planned to Titan, and we've actually landed there before. The Cassini-Huygens mission has this washing machine-sized spacecraft actually landed on the surface of Titan and broadcast back images from the surface of Titan. So we know we can do this, and there is a mission that's planned to launch 2027 called Dragonfly, which is basically a drone that can fly around the surface of Titan and study its chemistry, its physics, and possible signals of life. And I just want to move quickly through some of the technical and philosophical challenges associated with this. We have some tremendous opportunities to explore the solar system over the next few decades, but we need to better refine some of our concepts about life and habitability and life detection in looking at these environments, particularly under extreme conditions. One of which are the technical challenges. You don't have a full-on laboratory with all of the huge instruments and sensitivity in space a million miles away that you do here on Earth. And so finding ways to compartmentalize, miniaturize, and make autonomous some of these life detection approaches is really key. And it becomes more challenging as you try to go through an icy shell or try to land on the surface. Another issue is if we have models that say life could exist on these other planets and moons, are we potentially contaminating them with Earth life along the way? Or what do we need to do or consider along those lines? We've seen already that there are environments on Mars where Earth organisms in theory could exist, and we know we've landed things on the surface of Mars. So how big of a concern is that The JUICE mission I mentioned the other day is its planetary protection plan is to crash into Ganymede, which, as we've just described, has a subsurface ocean. So, is that a concern? Back in the 70s, when the Viking lander was being prepared, they would cook the whole thing, make a Viking casserole, right, as a way of sterilizing it before it was launched. This also presents challenges with regards to the technology and the engineering. Nowadays, NASA has these dedicated clean rooms where they carefully swab and do statistical calculations of the organisms and the biomass contained and do their best to minimize potential forward contamination as they're exploring the solar system. And then finally, how biased are we based upon what we know? We've already seen how the bias towards mesophilic organisms that have been well studied, influences our maps of habitability. But how biased are we based upon Earth life, based upon requiring that life requires liquid water or has DNA or undergoes evolutionary processes? Are there more generalizable criteria we can use to search for life that don't depend upon these things that we know from Earth-based life? So, certain aspects of people involved in astrobiology and space exploration are considering whether there are some more generalizable criteria that can define life from non-living processes in the search for life elsewhere. So hopefully through this talk you've seen that, that we still have a lot to learn about the environments and the life on Earth, which is why I had that in the title, the search for life on Earth and beyond, that there are habitable places elsewhere in the solar system. And in fact, we haven't even talked about extrasolar planets, which provide a whole nother order of magnitude more opportunities for habitable environments in the universe. I'll leave it at that. Here's a picture of Elvin diving into the abyss, and I'm happy to take any questions. Thank you.
With the ongoing loss of ice in our pools, do you anticipate the loss of organisms that we are still not even aware of that might be very localized? From one location to another, they might be different. And is it conceivable that as an ice mass melts, we will permanently lose some life forms?
I think that certainly is a possibility, but here's one thing to consider is that we've had past episodes of almost completely ice-covered Earth and past episodes of completely ice-free Earth. And so over the billions of years of Earth history and life on Earth, there have been these cycles before, and life persists. Or life, some life goes extinct, but a lot of life, particularly microbial life, persists and evolves as an adaptation for that. So I'd be less concerned about the loss of microscopic biodiversity in that sense, because it's part of typical Earth cycles. But certainly other organisms, multicellular life, could be impacted by that.
It was in one of your earlier slides, you showed sort of an adaptation to higher temperatures.
Yes.
And I just wondered, were those later ones due to a better ability or an increased ability to identify them? Or were they actually evolutions or adaptations?
Were the later ones in terms of as the temperature limit has gone up?
Had they adapted, or were they actually new ones that you identified?
So I think you're talking about the plot of year versus upper temperature limit, is that correct?
Yep.
Okay. So I think it's due to just persistence and creativity in terms of cultivation. So the strain 121, one of the upper temperature limit ones, was actually isolated by a person in microbiology department here in CNS, Kaz Kashefi, and he grew that using rust, basically, for the organism to breathe. And so that's an example of sort of using a creative new approach to identify these organisms. Another thing is to use high pressures, this is something we're doing in my lab, to squash organisms and study their pressure adaptations as well.
Okay, so that was a new one discovered.
Yes.
Do they adapt, though, as, if temperature is rising? some of them are adapting to the higher temperatures?
That's a really interesting thing to explore. So I don't know that anyone has really looked at that, but people are using experimental evolution to study, can in the lab, can you force organisms to adapt to new conditions? Yeah. And so that's a really fascinating area of research. In fact, that's something I'm excited, not that specific question, but that's some perspective I'm excited to integrate over the next few years in my own lab's research studies of these environments.
Based on your research, do you have any theories about where life on Earth originated? For example, in the deep sea near these vents?
Well, I mean, my perspective, because I've been there and because I was trained in these places, is in favor of hydrothermal vents, because it's not just hot, they're chemical reactors. They're fluxing material through the system. There's minerals all over the place that look like enzymatic reaction centers. So they're very dynamic systems that were protected from meteorite bombardment on the early Earth as well. So I personally lean in that direction. But the other important thing to consider is it wasn't just volcanoes or UV chemistry at the surface or warm little ponds. It was all of these things. And so, and it's all of these things on Earth today. The oceans and rivers and humans are all interconnected. So I think it may have required ingredients from each place to contribute to those early origins, but we don't know. Yeah, it's really cool. Except the first time you go down, when you come up, like the initiation thing is that people take your clothes, get them wet, put them in the freezer, and then they douse you with water when you come out of the submersible. So that's, yeah, it could be worse. It's fun. All right, thank you.
Well, I can say, Matt, I think I did find the place on Earth that's warmer than 122 degrees, and it's the stage. So, yeah, for anyone else coming up. So it is now my pleasure to introduce Angela Wilson, John A. Hanna Distinguished Professor, Nat Sy Associate Dean for Strategic Initiatives, and the immediate past president of the American Chemical Society.
Well, good morning, everyone. A key component of Nat Sy's undergraduate education experience is research, and we are committed to advancing knowledge by providing progressive undergraduate research opportunities. Whether it's developing research plans, writing proposals, conducting research, analyzing data, or presenting research results, these opportunities provide students with a deep understanding in a scientific area while gaining hands-on research experience. Undergraduate research is a great way for students to learn from faculty, from other researchers, including graduate students and other undergraduate students who share similar career interests and goals. Simply put, being involved in research greatly enhances a student's degree while providing them with the experience needed to make them competitive for graduate school or science careers. Recognizing the value of having these students communicate their research and campus experiences so that anyone can understand what it's like to be a student at MSU today, the college established the Dean's Research Scholar Program in 2012. The Dean's Research Scholars personify the high caliber of the college's student body and exemplify the traits of true Spartans, hardworking, intelligent, problem solvers, and we were honored to have these students represent Nat Sy this past year at events and speaking engagements. To help give us a little bit of a glimpse into this undergraduate research, here is Biomedical Laboratory Science Senior Sean Crowley. Sean, please join me.
All right, so welcome everybody, good morning. Today I'm going to talk about biological control and the fight against the emerald ash borer. So There we go. A little bit about me real quick. I'm going to be graduating in May with a major in biomedical laboratory science and a minor in sociology. I work as a head lifeguard and Red Cross instructor at a health club nearby. I volunteer at Sparrow Hospital, and I did my research with the U.S. Forestry Service and MSU Entomology under Dr. Toby Patrice and Dr. Therese Poland. Now, real quick, I do want to kind of just get out of the way. They're very similar words. So entomology, the study of bugs, etymology, history and origin of words. And I'd like to just sort of make that distinction real quick, just because when they do get mixed up, it bugs me in a way that I can't put into words. So, forgive that pun. So the big problem real quick, there's about 8 billion ash trees in the United States alone. And that guy in the middle, the emerald ash borer beetle, is responsible already for the death of over 40 million of those ash trees. Now, what we want to do is we want to make sure that 40 million doesn't turn into 8 billion. And so we're using biological controls to sort of stop that process. What is a biological control? So it's the use of another organism that we introduce into the system that controls the emerald ash borers for us. In our case, we use the Spathia scaleni, which is a really tiny wasp that we introduce, and it will go in and it will lay its own eggs inside of the emerald ash borer eggs, thus killing them and sort of controlling the spread of that infestation. And another good thing is just sort of it allows us to prevent the use of pesticides and chemicals. We can introduce another species into the system, and we don't have to sort of go in and use harmful chemicals that could hurt other things. This here is just a map of the United States. Every county in yellow is where we are seeing emerald ash borer infestations. So we are seeing it's mostly sort of Midwest and the East, but it is spreading south and west. And in this map, we see every red dot is going to be areas where biological control has been used to sort of fight that process. And up there you see right around mid-Michigan, all of those red dots right there is sort of where our lab worked and where we were going in and releasing the wasps. My role specifically, trap collection. So you see these big yellow traps in the top right corner, that's a pan trap. Inside is a sugary solution that attracts the insects. So we would go and hike through swamps and forests, through mosquitoes and everything, and collect these traps on a weekly basis. We would take them back to the lab and we would look at them under a microscope and sort of determine what we found in the area. And we were looking for those tiny wasps. We were trying to make sure that week after week we were still seeing them within the area that we released them, and that they were being established. Secondly, I worked with the DNA extraction of many of the insects that we found. Because we worked with the U.S. Forestry Service, we weren't only interested in the emerald ash borers and the wasps. We were looking at everything we found and sort of seeing, is there anything new? Is there anything that we didn't know about? Are there changes in anything? And so we did that DNA extraction, we sent it away to be sequenced. And then third, we did a lot of rearing of our own beetles. So that process looks a lot like this. You see the cups there, we have a whole bunch of ash, and ash trees, and we usually keep about 1 female with 2 males. And in between the coffee filter that you see on the top and the beetles, there's a fine black mesh. And so that black mesh simulates the inside of the bark that the beetles would naturally lay their eggs on. And so they'll climb up on that, and they'll lay them onto the undersurface of that coffee filter, which we can remove very easily and use those eggs in our research. So one of the big things that is important always is how do we know that what we're doing is working? So what we would do is we would go ahead and build these contraptions here. They are allowing us to scrape the bark off of the ash trees and minimize the loss of the bark shavings. So when you use the draw knife to do that, it kind of sprays all over the place. And so the box around that lets us catch everything that we're scraping off. What we do with that is we go and we sift it, and by sifting it, we're knocking all of the eggs off of the bark, and we're collecting all of the really small, tiny pieces. And what we do with those is we look at them under a microscope. That's about what it looks like, and we take the smallest paintbrushes you can imagine, and we go in a microscope, and we're sort of combing through these ash bark shavings. And what we're looking for is the eggs. So we're looking for the emerald ash borer eggs, and you can see they look a lot like bark. So it's difficult to go in and find them. But when you do, there's a way to sort of tell if they've been parasitized or not. That's when the wasp lays its eggs inside of the emerald ash borer eggs. So there's a little tiny black dot, and that tells us that what we have released in the area, the wasps that we released, are doing what we want them to do. Sort of what's at stake. So there are 16 species of ash trees, and we want to make sure that that number stays where it is. There's a ton of loss of habitat in areas where ash trees are. I mean, there's all sorts of biodiversity that we see around these trees, and destruction of property. They're beautiful trees, people see them in their yards, and we want to make sure that they stay around. And then lastly, we want to make sure that we're curbing the spread of infestation. Now, I'm sure you're wondering why a biomedical laboratory science student is hiking through forests and dealing with emerald ash borers. So I knew that I wanted to go to medical school, and so the first thing you read when that happens is, oh, make sure you get research, make sure you do this. And sort of the first thing that crossed my plate was the opportunity to engage in this research. And I thought, okay, why not? We'll see what it's like. And I made that snap decision. And it ended up getting me some really good experience with some DNA work. Also exposed me to anything from sample collection to being out in the field, anything along those regards. And it really taught me sort of what research is supposed to be about. So, for years growing up, everybody tells you, oh, science is about asking questions. It's about going out there and finding things. And taking the time and actually being able to go through and do that really did show me, wow, this is like the coolest thing ever. What's the answer to this? Or like, why are we doing it this way? And so by taking the time and doing that, it really promotes that. Thank you. Does anybody have any questions?
Good morning. I live north of Alpena, and we live up on a lake. And I got hundreds of dead ash trees already from your friend there. So, and what's the progress? Has there been any luck?
So I did know that by the time that I was finished with my research, we were seeing up to about 60% of the emerald ash borer eggs being parasitized and dying. I can't tell you the exact specific conditions that make that happen. That's one of the things that we're still working on, is sort of, what amount of light exposure, what temperature, when in the season to do it. And so there is promise, there's progress, but it's a long way off before it becomes perfect.
Yeah, they spread so fast. It was amazing.
Correct.
Right. Just one more real quick question. What's the weirdest thing you ever found in a trap?
We found a flying squirrel in one once. It was alive. It did not get hurt, but.
There's stand. Yep. That's a two-step process. Have you looked at the danger? There's always a danger when you introduce biological controls. And there's two questions. One is, how do you raise these little wasps? And how do you get enough of them? And secondly, how do you certify that they're only going to do what you want them to do and not start picking on other species?
Right, so we actually got the wasps from other labs that would rear them for us. So we got them in big coolers full of and little tiny containers that had about 100 of them per container. And because we were working with the U.S. Forestry Service, they have a ton of experience sort of dealing with the process and the life cycle of the wasps that we were releasing. And so we got confirmation and sort of permission from the U.S. Forestry Service. Okay, you can release them. You can release this many in this location. And so while we weren't necessarily dealing with sort of why is it safe for us to release these, we did make sure that we got the correct permission from the U.S. Forestry Service to do that.
I'm going to extend that question a little bit, because I always find these fascinating, these questions of biological control. We're thinking about it with mosquito populations because they transmit diseases. So we like ash trees, and so therefore we don't like emerald ash borers, and so we want to get rid of them. But is there any theoretical consideration in the way you think about this, or the lab you're working in, about what ash borers might be doing besides killing emerald ash trees, and maybe we don't want to eliminate them from the population? I mean, these are just theoretical considerations, and there's no answer, and we can argue about them. But we just saw this great talk about biodiversity, and are we worried about eliminating biodiversity of ash borers because we like ash trees? Things like that. I mean, again, no real answer, but what are your thoughts on that?
So they are an invasive species, and so I don't think we're ever going to get to the point where we're 100% just getting rid of them altogether. I think they're always going to be sort of present. I don't think the goal is to totally just eliminate the species at all, but we're trying to prevent the elimination of all of the ash species. So at the moment, our mindset, like you were telling us, is sort of on the fighting of the ash borers. I would be interested in research sort of surrounding the other things that they're doing in terms of their life cycle, if they're sort of helping other species, whatever. I'm not familiar with that, but that's a good question.
I've got a question you can answer and one you can't. I live right here locally, Delta Township. Am I able to participate at all in your study in terms of traps or recovery or any of that?
So I believe that if you were to contact the U.S. Forestry Service or any of the labs doing this research and just say, hey, I've got this area of land, there's all these ash trees, they are infested, if you want to come and do some of this research, you can. I don't believe there's a way to apply for, like, specifically, hey, please come fix my emerald ash borer problem, release some wasps in the area.
And the question you can't answer, you're finding the wasp in, I think you said 60% of the eggs?
Up to about 60%, depending on the conditions and sort of where we are.
And is that significant in terms of controlling the ash borer?
It's significant in controlling the spread based on previous methods. So we still have a long way to go before we're sort of stopping the borders, right? We're keeping them where they are. But it's, I mean, if you think about it, 60%, that's a whole lot less that are going on to have more eggs. So we're getting there. In the grand scheme of things, there is still a long way to go.
I'm curious about the mechanism that the eggs, the borer eggs in the tree, and how they actually damage the tree. What is it about the eggs and their presence that actually causes the tree to die?
So in the life cycle, and let me just go back. Oh, I don't have control anymore. One of the pictures you see all of the little lines that show up on the bark of the ash trees. They're called galleries. So it's pretty characteristic of a tree that has ash borers on them. So when they hatch, they grow into sort of the larva, and they will crawl through and they eat the inner layer of the bark, and that's sort of how the tree transports nutrients up and down. And so through that process, they're starving the tree, disconnecting it from the roots and the leaves, and that kills the tree.
Don't transport firewood.
Yeah, don't move firewood. It bugs me. I think we have a question over here.
You mentioned that the emerald ash borer was an invasive species. Are the wasps their natural enemy? So is the wasp a natural species here, or is it invasive too?
It is not. We brought it over from, I believe it was somewhere over in Asia. But obviously extensive research was done to make sure that it's not really affecting or targeting any other populations.
Thank you, Sean. That was a fantastic talk. Next, we will hear from Julia Ganz. Julia is a developmental biologist using zebrafish as her preferred research organism. During her career, her research has focused on the nervous system, spanning from the brain to the nervous system of the gut, the enteric nervous system. Julia joined the Department of Integrative Biology as an assistant professor in 2016. At Michigan State, she is researching the genes that regulate the formation of the enteric nervous system and what this tells us about diseases that affect our gut and digestion. Julia has contributed to over 25 publications and won prestigious awards, including recently a very large NSF Career Award. Please join me in welcoming Julia Ganz to the stage.
Hello, everyone. So thanks a lot for the introduction, and I'm really excited to talk to you about the brain and the gut and how it forms during development. So I'm starting you off with having you look at this movie. This is actually part of the gut of a zebrafish. And you can see that there are those really coordinated movements. And those are the movements that allow food to move through your gut and that are really important for digestion and uptake of nutrients. The other thing that I want to tell you about is why we use zebrafish, so this little striped fish that's up at the right corner, for research. So what basically the cool things are that this fish can and help us answer our questions. So if you think about the nervous system, you probably think about the central nervous system first, which is your brain and your spinal cord. But there's actually also the peripheral nervous system, which basically is every other part of the nervous system that's not your brain and your spinal cord. So the peripheral nervous system contains the enteric nervous system, which is your gut nervous system. And one thing that I want you to take away from this presentation also is you do have a nervous system in your gut, and it's really important. So your nervous system contains two important cell types, the neurons that regulate the activity of the nervous system, and glial cells, which have a variety of functions in the nervous system. They are also important for the activity of the nervous system. They support neurons and so on. The research in my lab is really interested in understanding how do those different types of neurons and glial cells form during development. Okay, so a little bit more details on what the enteric nervous system does. I told you it's important, and you should remember it. So because it's the gut nervous system, it is really important for gut movements. So it moves the food through your gut. Thereby, it's really important for you to digest your food, take up nutrients, absorb water, and so on. The nervous system of the gut also interacts with a lot of other cell types in the gut. So, for example, there's a lot of immune cells in your gut, and the enteric nervous system interacts with those immune cells, which is really relevant also for diseases that affect your gut, like inflammatory bowel disease. The enteric nervous system is also really important for intestinal barrier function. So if you think in your gut, even though it's inside of your body, it actually is kind of like an outside surface, right? Like things come in from the outside, go through your gut, and then go out the other way. So the enteric nervous system makes sure that there's a real good intestinal barrier function so that things that are inside in your gut, particularly the gut bacteria, don't get into the rest of your body, which would be pretty detrimental. The enteric nervous system, really last but not least, is also really important to make those bacteria in the gut happy. So we need bacteria in the gut. They're the gut microbiota. You might have heard about it. It's kind of becoming really prominent newsworthy thing that gut bacteria are really important for health of your gut and disease. So the enteric nervous system is really important to make the right environment so you have the right bacteria in your gut. So even though your enteric nervous system can function independently from your brain, it's actually the only part of the peripheral nervous system that does not need brain on, but you can take out the gut and it still moves. It is connected to the brain via the gut-brain axis. And just to give you a little bit of the scale of your enteric nervous system, it actually has as many neurons as your spinal cord. So it's a really large part of the peripheral nervous system. Okay, so how do you make a nervous system during development? So this is what my lab is really interested in. And I'm just going to break this down just generally how you make an organ, and I'm going to give you this with the ENS as an example. So to make an organ or a specific cell type during development, you start out with a stem cell population. Those stem cells then multiply, and they give rise to other cell types that are a little bit more going along a certain path, for example, towards neurons. Those cells then multiply also, and at some point, sort of through different steps during development, you get different types of ENS neurons. So now you can ask yourself, how is this process regulated? Like, how do those stem cells know that they should give rise to more stem cells or make a neuron? And the answer to this is that they receive specific instructions. And those instructions are within the DNA that is within each cell of the body. So I'm sure you've heard about DNA and you've heard about the genome. So you can think of the genome as a collection of instruction manuals that basically tell cells what to be and what to do. So within the genome, you have one instruction manual, which is the gene. So a gene is really kind of the unit that is important for telling a cell what to do. This gene is regulated by so-called gene regulatory elements, or GREs, And both those terms, genes and GREs, are going to be relevant throughout the rest of my presentation, so kind of just keep that in mind. The gene regulatory elements are just pieces of DNA that you can think of as on and off switches. So basically, they tell a gene to be on, or they switch off the gene activity. Genes generate proteins, and during development, specific proteins are really important to help those cell fate decisions that I told you about. So they basically tell stem cells, okay, you go on and you become a neuron, or you become another stem cell, so you can continue multiplying. So if you go back to the schematic that I told you, showing all those different steps going from a stem cell to a neuron, you see all those arrows, and those are the different steps. And those arrows are basically filled by gene activity. So different genes are important for different steps of the way. So what happens if those instruction manuals break or go wrong? You can probably guess it. They lead to diseases. There's a lot of different diseases that affect the enteric nervous system. And the work of our lab is, as Phil pointed out, is fundamental research, understanding genes and what they do normally and what happens if they break and don't do the normal job anymore. But I'm just going to give you a couple of flavors what ENS diseases are like. One of the most prominent diseases in the enteric nervous system is called Hirschsprung disease. It's characterized by an absence of neurons at the end of the gut. It's diagnosed mostly in infants because they can't pass stool through their gut because they lack neurons. So the food can't move through the gut. The gut gets really large. This is also why this is called megacolon. And it's actually a really complicated disease because different mutations in different genes, and a mutation is basically just a change in DNA sequence that renders this gene non-functional. So there are different mutations in different genes. One that's really important is the red gene, and I'm bringing this up specifically because I'm going to come back to that in my presentation later. So those are sort of typical ENS diseases, but recently there's really been important developments that think that a lot of gastrointestinal symptoms that you see might be actually linked to altered development of the enteric nervous system. So for example, in neurodevelopmental disorders like autism spectrum disorder, there's a really high prevalence of gastrointestinal symptoms. And so recent work has shown that it might be partially due to abnormal development of the enteric nervous system. So one of those challenges with diagnosing and treating ENS diseases is that we know very little about the genes that control the formation of the enteric nervous system. So because of this, we really have too few candidate genes to look at in different patient populations. So there's a big problem that the genetic basis of ENS disease is often not known, and this is why we need to find out more about what genes regulate the development of the enteric nervous system. Because those diseases are really impactful, we don't really have good treatment options at the moment except treating the symptoms. So for a Hirschsprung disease, for example, you surgically remove that part of the gut that lacks neurons, which is fantastic that we can do this because it saves the life of the patient. But there are really a lot of problems even afterwards. There's still gastrointestinal problems, and there's things like life-threatening inflammatory reactions. So there's a really big push to develop more sort of reparative treatment strategies. There are two main approaches that are being evaluated. One is transplanting stem cells, basically putting in stem cells into the gut so they can make new neurons and glial cells, or regenerative approaches to basically try to activate stem cells that are still sitting within the gut and basically ask them to make new neurons. For both of those, I really strongly believe that we really need to have a very good understanding how this process works normally during development. Because if you want to use stem cells or if you want to use regeneration, you need to understand how this works normally, and then you can sort of start to apply this. And again, we're doing fundamental research. We're both looking at development and regeneration of the enteric nervous system. And I think this is really sort of an important basis to be able to use therapeutic treatments in the future. All right, so told you a lot about genes and gene regulatory elements, but how can we actually test their function? There's a lot of different ways you can do this, But one of the sort of most powerful things that have just been recently coming about, that I'm sure you also heard about, is to use what's called CRISPR genome editing or gene editing. So how does this work? So the nice thing with CRISPR is that it's pretty straightforward to do, and the way that it actually alters gene function is that you use two proteins, those are the Cas9 proteins. They cut DNA, so they basically make a cut in the gene. If you use two of them at two different sites, they will cut out part of the gene, and the gene will not be functional anymore. So if you have this non-functional gene, and what this is really doing is just making a mutation in the gene, you don't get a protein, and now we can ask, how does this change development? Like, what happens if you don't have this gene functional anymore? So just to give you an example, I talked about the red gene and its importance for Hirschsprung disease. So if you were to make a mutation in the red gene or a mutation in the gene regulatory element of the red gene, you would get no or less red protein. This would lead to no or fewer ENS neurons in the terminal region of the gut, and this can lead to Hirschsprung disease. So one thing that I want to emphasize is I deliberately mentioned the gene regulatory element here because a lot of mutations that have been connected to human diseases actually sit in gene regulatory elements and not in the gene body. They can happen in both, but they're actually very prevalent in gene regulatory elements. And this is why it's so important that we look both at gene regulatory elements and at genes to really get a full understanding of how development works, but also how this might impact disease. All right, so the question really that we are getting at is what genes and what gene regulatory elements regulate ENS development. So that should be all, we should be all set on that. But why do we use zebrafish to do this? You might have seen zebrafish in aquariums. They're like this little striped fish. They're really easy to keep and breed. This is why we like them. But they also have really cool other advantages that I want to tell you about. So one is they have external development. So the eggs are laid outside of the mom, so we can look at a developing embryo right off the bat. They also develop really rapidly, so that's nice. They have a really high number of offspring. One pair of zebrafish can give rise to hundreds of eggs, so we have a lot of embryos to work with. And the really, really cool thing is that the embryos are transparent or optically clear. So you can look at a live animal during development and just see what's going on without having to do anything else. And you've seen this movie here on the side, which is an early developing zebrafish embryo. You can see they're optically clear, you can kind of see right through the embryo. But the other thing I want to emphasize is, again, you can already, at the end of this movie, you have the head on the top, there's like an eye forming, a body is forming, a tail is forming, and this is after 19 hours post-fertilization, which means in less than a day, you have an embryo that's clearly identifiable, you can have with a head and like a lot of important structures that are formed. So this also works for the enteric nervous system. Now we're looking at a zebrafish larvae at 6 days post-fertilization. So a 6-day-old one, the head is to the left, you see the eye on the left side, there's a swim bladder, and the gut is just really nicely accessible in the zebrafish. We can look at the enteric nervous system in a live animal. So you can see in the middle panel ENS neurons that are basically identified with a green fluorescent protein. On every green dot is a neuron, and you can just see them right there. If you look at the close-up, you can identify single cells, you can see their processes. The other important point for us is that they have conserved genetics to mammals, including humans. So about 70% of human genes have an equivalent in zebrafish, and there's many shared genes during ENS development. And I'm going to give you 2 examples just to kind of exemplify this point. If you look at a mutation in SOX10, which is an important gene for ENS development, we have a wild-type zebrafish on top, and we have a SOX10 mutant zebrafish on the bottom. And SOX10 mutant just means the SOX10 gene is non-functional in this zebrafish. So you can see that there's a lack of pigmentation except for the eye. And if you look in the cross-section through the gut, you can see in the wild type, you have the brown cells around the gut. That's ENS neurons. And in the SOX10 mutant, those ENS neurons are gone. If you now look at a mouse, we see exactly the same phenotypes. We have pigmentation phenotypes, and we have ENS phenotypes, meaning that in the wild type, you have those black cells where the arrow points at. In the SOX10 mutant gut, you do not have those cells anymore. So the phenotypes are the same. Another example is the red gene. I told you this is important for ENS development and for disease. Now we kind of look at a close-up view of a wild-type zebrafish gut. All the white dots are ENS neurons. And below you have a red mutant where the red gene is not functional. And you can see there are no white dots anymore. The same is true if you look at a mouse that is mutant for red. You have the wild-type gut on top. You see those brown lines. Those are the process of the ENS neurons. And below, you do not have those brown lines anymore. So there's conservation pathways, meaning what we find out in zebrafish is really relevant for mammals, including humans. The other nice thing is that zebrafish are really well suitable for testing human candidate genes using CRISPR and for functionally testing gene regulatory elements. So when I arrived at MSU, I had the privilege to build a really large zebrafish facility on campus, which serves 2 labs. So we started out with 8 fish racks and 1 quarantine. This is how it looks like now, much more filled up. And we now have about 8,000 zebrafish in our facility and about 60 different genetic lines. What do we do with those fish? We're going to start with looking at how and what genes regulate ENS development. So I'm just going to show you a small flavor of the work that we do. And this is work that was spearheaded by postdoc in the lab Annie and 4 fantastic MSU undergraduate students. And I want to really emphasize that this is also a really cool aspect of this research, that it is accessible to undergraduates. And I'm really, really passionate about having undergrads in the lab and kind of giving them this research experience because I do think this is really important for them. All right, so how can we use CRISPR to figure out which genes regulate ENS development? So to do CRISPR in zebrafish, you inject the CRISPR components in the egg. Then you raise those embryos to 6 days. And now we can look at the enteric nervous system. So at the top, you have sort of a schematic of a zebrafish larvae. The gut is in yellow. And below, the dotted line outlines the gut. You can see all those green dots, right? Those are ENS neurons. And below, you see an injected zebrafish larval gut. And you can see there are a lot less green dots. So clearly, there are fewer ENS neurons. To make this more efficient, we actually target several genes in one pool, and we then look for changes in neuron numbers. Doing this screening approach, we started with 4 pools of genes. 3 of them contained fewer zebrafish larvae with fewer ENS neurons. And now we have a really nice set of genes that affect ENS neuron numbers that we can now work further with to figure out what is exactly going on, how do they affect ENS development, and do those actually also connect to ENS diseases? One other thing we wanted to look at is, can we see that if in those injected larvae, we also see effects on gut function? I told you one of the most important parts of the functions of the ENS is that they regulate gut movements. So how can we look at this in zebrafish? We can use a very straightforward and nice assay, which is to just feed the zebrafish larvae freshness labeled food, which means the food is going to glow green under the microscope. And yeah, it chose the color green for a purpose for this presentation. Not just go green, but glow green is our motto. So you feed them freshness labeled food, they eat it up, you select zebrafish that eat, transfer them into a clean dish, and then you look if they have cleared the food out of their gut or not. So on the top, you have a wild-type zebrafish control. You can see the gut is empty. Below, you have a positive control, which is the SOX10 mutant. And you probably remember that those mutants do not have ENS neurons, so they can't move food through their gut. And you can see this green stuff is stuck in that front part of the gut. If we now look at one of our injected zebrafish larvae, you can see that they also have the food stuck in the front part of the gut. So if we quantify this and look at the percent of larvae that have cleared their guts, SOX10 mutants and the zebrafish larvae in which we target a specific gene clearly have a lot fewer larvae that have cleared food through their gut. And this indicates that there's really also an impact not only in ENS neuron numbers, but also on gut function. Okay, so I just want to show you briefly a case study which we recently actually got more funding for, so we can continue this work, is can we now actually also look at human disease candidate genes? And we were focusing on candidate genes for autism spectrum disorder. So just as a reminder, individuals with autism spectrum disorder have a really high prevalence of gastrointestinal symptoms. Between 30 to 70% have gastrointestinal problems like constipation, abdominal pain, and diarrhea. And one challenge with autism spectrum disorder, there's actually a huge amount of genes that are connected to ASD. It's hundreds of genes. If you have a lot less, we could just test all of them and look, is there a connection to ENS development? But we have hundreds of them, and even though things are efficient in zebrafish, it's not quite that efficient yet. So I've teamed up with Dr. Arjun Krishnan, which is at Colorado Anschutz, to see can we sort of prioritize genes that are likely affecting ENS development. So we've developed an integrative computational experimental approach to do just that. Dr. Krishnan's lab is the computational side of things. So he's using gene expression data, gene network analysis, and other informatic approaches to find a list of sort of prioritized ASD and ENS genes. And we are now testing those in zebrafish, and we did a proof of principle screen. Annie again led this together with another undergrad student. And out of the genes that we tested, we were really excited to see that there's a good number of them that affect ENS neuron numbers. So it's really nice to see that there is a potential connection between some of the autism spectrum disorder genes and abnormal ENS development, so that we can potentially make this link to better understanding the gastrointestinal symptom prevalence in autism spectrum disorder. All right. So now, in the last part, I'm going to tell you a little bit about gene regulatory elements and how we find those and how we can test them in zebrafish. So how do we find gene regulatory elements? That's a great question. So let's see. So when I showed you the DNA before, I showed you basically like a strand of DNA. This is not how DNA actually is in the cell. DNA is actually wound up tightly around specific proteins, and they come in two different configurations. Either they're really tightly wound up. This is what's called closed chromatin. Chromatin is basically the term for the DNA that's wound up on those proteins. And if it's closed, it's really inaccessible for any protein that would like to kind of get to this piece of the DNA. Or the DNA can be not bound on proteins, not tightly wound up. If it's more open, proteins will actually be able to bind, and gene regulatory elements need proteins to bind to them and activate them. So there is a method that's called acid-for-transposase accessible chromatin, or ATAC-seq, which allows us to tag those open regions of chromatin and to identify the DNA sequences that those are. So using this method, we can find the regions of open chromatin, which are potential gene regulatory elements. So we did this for ENS cells and surrounding gut cells. And the data that we get looks like this, and I'm going to walk you through that. So on the top part, you have the red gene. So we are coming back to red, of course, because it's an important gene. And below, you see those little peaks, either green or gray. And below the peak, you kind of see sort of a square box. If you have a peak and there's a box underneath it, this means that there's a region of open chromatin, and this means that this DNA sequence is a potential gene regulatory element. So you can already see that those peaks are kind of distributed differently around the red gene in those two different cell populations. And what was really nice to see is that one of them overlapped with an already known gene regulatory element of red. So we knew we were on the right way, like our data seems to be making sense. And then there are more that haven't been tested before that are specifically found within the ENS cells. There are some that are shared between the ENS and surrounding gut cells, and there are some that seem to be gut specific. So the other very cool thing about this data was that some of the gene regulatory elements actually overlap with sites where we have found human disease mutations of Hirschsprung disease. So this is exciting. If those are now really gene regulatory elements, we can test them in zebrafish and figure out more about the connection between Hirschsprung disease mutations and how they might potentially affect gene function. So how do we test those gene regulatory elements in zebrafish? We take a gene regulatory element and we hook it up to a gene that we can easily see under a microscope, or we can see the gene product under a microscope. So we hook it up, for example, to green fluorescent protein gene. then that green fluorescent protein gene, if it's active, makes green fluorescent protein, which is a green protein that we can see under a microscope. We inject those into zebrafish embryos, and remember, we have hundreds of embryos that we can inject in, so we have a really nice amount of embryos we can look at and work with. Once we've injected them, we're going to grow them up, and we're going to look, are the green fluorescent protein cells the same cells that we know express red gene and make red protein? So we can basically compare the expression patterns to known expression patterns of red. All right. So with this, I hope I've shown you or convinced you that looking at development in general, but also looking at ENS development, is a really good way to understand how you make a functional ENS. I've not had a chance to talk about regeneration. Zebrafish are very regenerative, so this is a new research area in my lab, and I'm very excited to get going with that. And both those research directions will also help to inform and build new models for human gut diseases. So with this, I would like to acknowledge people in my lab, funding sources, and I want to give a specific shout out also to the MSU GI group. I just want to emphasize MSU is actually really big in ENS research. It's very big in gut research. We have a lot of labs working on different aspects of ENS biology and gut research. So MSU is a good place to be if you're interested in your brain and the gut. I'm happy to take questions.
Hi, my question is about one of your earlier slides. I'm recently retired family physician, and over the years you'd hear patients talk about colon cleanses and enemas, and the early slide talked about the importance of the ENS and the microbiota. I wonder if you have any thoughts about good or bad, or maybe not be real scientific, but I remember colorectal surgeon told me once, said it's not normal to clear out the gut bacteria.
That is a great question. And of course, you're starting me off with like a question where I'm not going to be able to really give a like yes or no answer. So I think it depends a little bit. I mean, overall, the composition of your microbiota is really important. And if things change in your gut, this will impact what bacteria you have in the gut, and this does impact disease. In terms of cleanses, I mean, in some way you could view them as sometimes helpful to kind of, if you kind of just want to sort of clean it through. The challenge is really that, you then sort of need to repopulate your gut microbiota sort of the right way. I think probably the bigger impacts on your gut microbiota are sort of more like the things you sort of do on a regular basis. I mean, the things that you eat impact what bacteria you have in the gut. And again, and also sort of how your gut actually functions in general with the ENS function. So I mean, it's always hard to say with gut microbiota, like, what is going to be the best way moving forward, because it is a complex system and we don't understand fully how it's formed and kind of set up. And we don't even fully know, like, what's the problem first? Like, is it because the gut isn't working, your microbiota changes, or is the microbiota changing, and this then affects how your gut functions? And it's probably both, because those systems are tightly interconnected. They talk to each other all the time. And your gut can really take up a lot of, also the ENS actually receives a lot of information from the gut microbiota.
So this is a different question. With the ENS, how much effect is there on inflammatory bowel disease? And is some of your research working with the GI department here involved with that?
Yeah, so it's kind of along the same lines. Because the gut has all those cell types that communicate with each other, looking at the cross effects of inflammation, how this affects ENS function and how ENS function. ENS function definitely impacts inflammatory responses in the gut. So figuring out how those two systems work together and all the information that kind of goes back and forth is really important. We don't focus as much on inflammatory bowel disease ourselves, but there's other research on campus that really kind of looks at both neuronal and impact of the ENS glia on how inflammation comes about.
I can't stand, so I won't. But my question actually was kind of about when you were talking about identifying genes related to autism within the zebrafish and their development of their ENS, you can't actually phenotypically define whether or not the fish themselves have the development of autism as well, right? You're just looking for the genes themselves?
So there are actually some phenotypic or kind of behavioral assays you can do in zebrafish that mimic some aspects of the sort of more brain phenotypes, or phenotypes is the wrong word, like the brain manifestations of autism spectrum disorder. So you can connect this. There are zebrafish models also for autism spectrum disorder regarding the central nervous system. We really focus on the enteric nervous system. So that's kind of our main focus, to figure out are genes that are connected to autism spectrum disorder also impacting development of the enteric nervous system. But you can also use zebrafish for that, for sort of this both effects on the brain and the gut. And that's actually an excellent question. I mean, this is really what's sort of coming up at the moment in the research with autism spectrum disorder. This question is, do those genes just affect the development of the brain and the enteric nervous system? Are those things separate? Is there maybe a connection also if your enteric nervous system doesn't develop properly? There's a connection between the brain and the gut. Could this also potentially affect brain development? So also there, the interconnectedness actually makes it really interesting and, of course, also challenging, as always.
And then one other question, if I may. Could you touch a little bit on what your lab's doing in terms of the regenerative properties related to zebrafish? I know you just talked about it very, very briefly, but it seems really cool.
How much time do we have? Not just kidding. So zebrafish can regenerate their nervous system really well. So you can injure the brain or the spinal cord, you can transect the spinal cord, and it grows back. No one has really looked much in the enteric nervous system, but the bets are on that they're going to regenerate the enteric nervous system really well. Just for context, we do not. So, I mean, we don't regenerate our spinal cord, our brain. We also don't really regenerate our enteric nervous system. So there's a little bit of stuff going on, but not really. So the idea really is that if you look at an animal model that can regenerate really well, we basically want to figure out how it does that. And at some point then also figure out what is different to us and to mammals. Like, why can they and we cannot? Like, is there something missing in us? Or is it just that zebrafish have something in addition? Is there a different cellular risk? There's just different way that this could go about. But the idea is to first understand it in an animal that can do it and figure out how that works, both on genes and cells and so on, and then go look in mammals and see what's different. And of course, hopefully also at some point then use this for informing therapeutic approaches or kind of seeing what barriers do we actually need to overcome.
Hello and welcome back. I hope everybody had a great break. One housekeeping item before we get started. If you do have a question, please wait for them to bring you a microphone because the people online will not be able to hear the question if you don't have a mic. So I am pleased to introduce Madeline Russell. She's a senior microbiology major who will tell us a little bit more about her research. Thank you, Madeline.
Do we have the clicker? We seem to have lost. There we go. Thank you.
Yep.
Hi, everyone. My name is Madeline. I'm going to be talking today about the infant gut microbiome, and I think I'm actually very lucky to be going second to last in this because we've already talked about the gut and we've already talked about bacteria in unique environments, and we've also talked about genetics a little bit, and all three of those things are going to be covered in my presentation as well. So I'm going to be talking about myself a little bit. So just as some background knowledge about me, I'm a senior majoring in microbiology. I'll be graduating this semester with both my bachelor's and master's in microbiology in 4 years. I'm originally from Marshall, Michigan, which is about an hour south of here. Additionally, I'm the president of 2 clubs on campus, the MSU Irish Dance Club and the Honors College Mosaic Club, which is a multicultural organization. And because I'm sure it's a burning question, the MSU Irish Dance Club had a competition a few weeks ago That's me standing on 2 legs, and unfortunately at the end of that competition I tore my Achilles, which is why I'm in the boot and the crutches today. It's okay, I can walk in a week, so I'm very excited about it. But just to tell you a little bit about that. Additionally, this year I was on the MSU homecoming court, which is a couple of those pictures, and I studied abroad in Germany this past summer. And I've worked for 4 years in the Comstock lab. I started off as a professorial assistant, which is just a scholarship that you're given as an incoming honors college student, and then moved on to pursuing my master's in her lab. So to talk about the microbiome a little bit, or just microbiology in general, I had no idea what it was when I was 18 when I applied to Michigan State University and chose it as my major. I'm really, really glad that I did, because I've come to love research and learning more about the gut microbiome in general. But just to talk about the gut microbiome, we heard a great presentation earlier about kind of the gut in general, and talking about the development of the gut. And part of why the gut microbiome is so interesting is that there's a lot of things that impact the actual presence of bacteria in your gut. So if you didn't know, this is me spoiling it for you, there's thousands of bacteria living in your gut at a given time. Most of them are good, but some of them are bad. And we can influence the gut microbiome and the composition of the gut microbiome through a variety of factors. In my lab, we tend to look at the infant gut microbiome and how a mom's lifestyle factors such as pre-pregnancy weight or obesity impact the development of her child's gut. So 2 of the main things that influence it are stress, be that exercise, metabolic, physiological, or diet. So I work in a human nutrition lab, like I said before, and so we look a lot at breastfeeding and how that influences the development of the certain types of bacteria that are present in someone's gut. And so, in turn, the gut microbiome also influences us. It leads to the development of a lot of diseases over time. We talked a little bit earlier about gut diseases, and that's one thing that if you have a bad composition of certain types of bacteria in your gut, it can lead to those types of diseases. Additionally, it can influence obesity, liver disease, diabetes, etc. And disruption of that gut is kind of what we want to prevent. So I'm sure all of you have heard about probiotics and other types of tools you can take to kind of change the composition of your gut microbiome. This is my personal plug that you should probably just eat a lot of yogurt, kombucha, that kind of a thing. But there's a lot of research that's being done about this, but we still don't know a lot about it as well. So I'm going to kind of walk us through just some of the 3 research projects that I've worked on throughout my time at MSU, starting with the project that I worked on for my first 2 years of college, was about studying RSV. And RSV is called respiratory syncytial virus. We've all been exposed to RSV within the first 2 years of life. However, it's typically a very mild disease, but in infants, it can result in sometimes in a really deadly and sometimes deadly, but also can result in like asthma and other long-term health complications. And so when I joined the lab, it was right before COVID, and then when COVID hit, I kind of was looking for a project to do, and we'd already done the DNA analysis on all of these samples that we had collected from a pediatric intensive care unit in Helen DeVos Children's Hospital in Grand Rapids. So I pretty much just had to go in and do the actual data analysis on our study. And so the objective of our study was to characterize, which just means to look at the different types of bacteria present and how much bacteria was present in the gut of these sick infants with RSV versus control infants. And so with that, overall, what we found was that RSV patients had very different gut bacterial communities than those of control infants, which sounds intuitive, but this wasn't something that had ever been looked at before. We had never really looked at how the composition of the gut microbiota is changed over time within sick infants, with RSV specifically. And this also came at a really interesting time because, like I said, this research was published right after the COVID pandemic. And so this has also been really interesting in the past year because RSV has come back full force this year. And if any of you have young grandchildren or children, there's been an increase in hospitalizations. So this research has been really interesting in the past year because it's gotten a lot more notice, a lot more traction on the website that it's published in, just because people are interested to see, okay, what types of bacteria are being associated with these sick infants, how can we combat them with different antibiotics, et cetera. And so just to kind of walk you through this graph, this is called a principal coordinate analysis. The main thing to take away from this essentially is just that the This is talking about variants. So each of these little dots represents a specific individual. The red dots are the sick babies, and the black dots are the control infants. And the separation, like how far away they are, means that they're more different. So those two ellipses that you're looking at are based on the centroids of the two different communities. So as you can see, the black dots are centering pretty far away, and they're also associated with Porphyromonas and Prevotella, which are two types of commensal or helpful bacteria in the gut. And so overall, this research just kind of was a preliminary look at how we can talk about how infants within intensive care units react to severe respiratory illnesses. And so after I finished this project, I think this research we published last January, the following summer, so this past summer, between my junior and my senior year, I took a detour to Germany. I was awarded a Dad Rise Fellowship, which is essentially just the German version of a Fulbright Scholarship. Not quite, because the Fulbright Scholarship is the Fulbright Scholarship, but that's the closest thing that I can amount it to. So I spent 12 weeks as a Dad Rise Research Fellow at the University of Dusseldorf. It was an incredible opportunity. I also learned that you don't have to speak any German in order to go be a research intern at the University of Dusseldorf. All I had to really know was how to order coffee, which was great. And so what we were doing this past summer was we were looking for novel genes related to diabetes in a female mouse model, which is also what these words say up here. But it's way more complicated and it doesn't really make sense. So essentially that's what we were looking for, and we were able to identify and replicate specific genes that they had identified in a male mouse model. The reason they were interested in a female mouse model is because a lot of times female animal studies are relatively underdone. A lot of the studies that we look at are predominantly in male species, so it was really interesting to replicate those findings within a female mouse model because obviously they've been understudied. And so I just kind of wanted to walk you guys through the process of what I did. So I talked about with RSV, I was mostly doing computational analysis in R, which I really enjoyed. And then this past summer, my job was to essentially isolate something called RNA from the adipose tissue or the fat tissue of these mice. They were already dead. They'd been long dead before I got there. But so I spent the first 12 weeks just taking 300 samples through this process and then doing computational analysis on them. And it takes like the full 12 weeks to do this. And so what it looks like is first you take the sample and you prepare it to extract the RNA. And so when we talk about DNA, RNA, and proteins, you start with kind of this code of DNA, and then the blueprint is kind of RNA that comes from the DNA, and then eventually you produce the protein. And so what I was looking at was the RNA, which kind of in a weird situation, you have to actually convert it into cDNA in order to do a specific type of analysis on it. And then you do something called qPCR, which basically looks at the fluorescence of the genes themselves. So I didn't actually include the data here from this study just because I'm gonna talk a little bit more about like the research that I've been doing with my master's, but I just wanted to show this to show how involved this kind of process is in order to actually learn about these different genes related to diabetes. So it was a really great opportunity and I had a lot of fun. And also the College of Natural Science helped me afford to be able to go on this with a scholarship. And so just overall very grateful to them. And so then moving on to kind of what I was interested in when I came back. I came back last spring semester, I was talking to my professor and I was like, I really don't want to graduate yet. What can I do in the lab? I want to stay for another year. I do not want to finish in 3 years. Like, please, just what can I do? And she was like, well, I have this really cool project related to antimicrobial resistance. And I was like, what's antimicrobial resistance? I've never heard of that before. And essentially it's when bacteria or fungi or viruses become no longer susceptible to drugs that they were once able to be treated with. So just to kind of give you a diagram of that, in this first diagram, I have all of these bacteria, right? They're treated with antibiotics. There's gonna be a couple of bacteria that survive. You know, like when you use hand sanitizer and it says it kills 99.99% of germs? This is what's left, which is kind of scary because these bacteria are now resistant when you treat them with antibiotics again. They're resistant, they're still there, and because of the nature of bacteria, they can potentially spread their genes through something called horizontal gene transfer to other types of bacteria. And so that's why antibiotic resistance is so interesting. It's an emerging, really dangerous problem around the globe. We know that it's increasing every year, and also what we've found preliminarily, is that these genes are being transferred from mothers to the gut microbial communities of their infants. And we don't know if that's happening not during pregnancy, because that's typically not, it's typically a sterile environment, but we think that it's happening either through the way that the babies are born or through the antibiotics that the moms are taking during pregnancy. And so that's kind of what this study is looking at. We wanted to characterize, okay, what types of antibiotics are moms taking that contribute to antimicrobial resistance in their gut, and how does that lead to antimicrobial resistance in the gut of their infants? And so kind of the first, just doing the preliminary analysis on this, some of the things that we found is that infants' resistomes, which is essentially just all of the antimicrobial resistance genes that are present in the gut of their bacteria, within their gut, within the bacteria, we found that they had a greater abundance of specific antimicrobial resistance genes by antibiotic class. So what that means, for example, is we all have heard of tetracycline, right? And so what this graph is showing is that the orange box plots represent the infants and the blue box plots represent the mothers, and that's a sum of all of their antimicrobial resistance genes. And what we found overall is that for most of these classes, infants were higher, which is really concerning because it's like, why are they higher? Why are infants having a greater population of these antimicrobial resistance genes, especially since they've never had antibiotics before? And so each of these blue circles with the blue and the gray represents a matched pair of a mom and their baby. And so what we see is that maternal is in blue. So that means all of the antimicrobial resistance genes that are coming from the mom, and then gray represents all of the ones that we don't know where they're coming from. And so on average, the moms and their infants shared about 60% of their antimicrobial resistance genes, which doesn't prove that they're coming from the mom specifically, but it's showing that there's a lot that's shared between them. And so then finally, kind of going back to the same plot that we saw before, there was over 200 infants included in this study. I think we had 235 infants and 99 mothers. And then of those, we had about 33 matched pairs. And we saw that overall, the bacterial communities were very different. These two graphs that were very similar to the ones that we looked at before, just a lot more infants. That RSV study only had like 20 infants, and this one obviously has a lot more. And so what we just saw is that overall they were clustering very differently, and that infants had very different resistome communities from their mothers. And so then to summarize just everything that I talked about, for my first 2 years, I characterized the gut microbial communities of those infants with RSV. Then I went to Germany and was able to look at genes related to diabetes. And then finally, for my master's program, I've been able to talk about antimicrobial resistance. And so just to summarize the next steps for me, I'm graduating in 2 weeks, which is kind of crazy to say. After that, I'll be applying to medical school and continuing to work on research for the next year. And then finally, my ultimate goal is to just kind of make advancements in medicine to that work to solve systemic inequities in health. And with this picture, this is a picture I took on my very first week at MSU, and I'm going into my very last week at MSU as we speak. And so I just think that this is an embodiment of Spartan's will, and I'm so glad that I got to spend my time here with the College of Natural Science and at MSU. So thank you guys so much. Are there any questions?
And they get quiet.
Hi, Madeline. What are you going to miss most about Michigan State and being on campus?
Oh, that's so hard. I'm gonna say I'm gonna miss running on campus, which is really funny because I can't right now. But yeah, running on campus was probably my favorite thing. And then on top of that, just the community of MSU. I think that, as all of you know, the alumni and just the students and the professors, it's really a home here at MSU. So I think that that's gonna be one of the things that I miss most about being here.
Hi, Madeline. Congratulations on your graduation, by the way. What are you going to do when you become famous?
Oh, geez, I don't want to be famous. I just want to help people and like maybe be a physician. That'd be awesome. But thank you.
Thank you, Manny. You're a star, and we love having you in the department. So I missed what those maternal infant cohorts, were those were all healthy, just come to the hospital sort of patients, or were they? Was there anything about the history of those cohorts that sort of led you to that cohort as a study group?
Yeah, so our cohort was actually it's an NIH, it's a really large NIH study called the Environmental Children's Health Outcome, the ECHO study, and they have hundreds of thousands of babies across the nation, and specifically there's a cohort that's triangulated between us, Ann Arbor, and Traverse City with 6 different hospital systems in Michigan, and we have access to essentially, I think there was over 600 infants that participated in this study at different time points. The ones that we had data for at 3 months was 235. And so part of what was interesting about the study is, one, it is based in Michigan. So these are hospitals all across Michigan that come from different demographics as well. Like we have some hospitals that are predominantly white and very wealthy, or some hospitals that are predominantly white and very poor, or black and very poor, or more of a mixed population. And so that was another thing that I didn't really talk about today, but that's been interesting for my master's research, is looking at how the gut microbiome looks very different across different socioeconomic and demographics factors as well. So great question, Dr. Dorita.
Thank you very much.
Pardon?
Where are you going to med school?
That is the $1 million question. I'll have to I'll apply, and wherever lets me in the next year, because it takes a full year. So I'll apply this upcoming June, and then I'll have a year to figure out where I'm going. So I'd love to stay in Michigan. I love the state of Michigan. So
the resistomes, there's a real positive correlation with the infant and maternal resistomes. You didn't speak of it, but there must have been a control that you had that was not an infant-mother matchup to be able to identify the significance of that.
yeah, so we had a lot of infants and mothers that were the matched pairs, right? And then we also had the control of, we had some mothers that didn't have, they were still samples that were taken during their third trimester of pregnancy, but not looking at their exact infants, right? So we did something called, I didn't include it just because it's a little bit difficult to explain, but something called a kernel density dissimilarity plot analysis. And yeah, I know it's a mouthful, and it's not about popcorn either, which is disappointing. But so anyway, it looks at basically the different communities, and it compares it by, okay, these are the moms and babies that are matched. These are moms that are just comparing random moms to random infants, and then comparing random infants to each other, or moms to each other. So you're right, that's a good thing to point out, is that I didn't include that in here, but we do look at those differences too, to see, okay, are we truly seeing this difference between moms and their own infants? So, yeah. Awesome. Good. Okay, great. Thank you guys so much. Comes the precarious part of me. You can come up first. That works.
Thank you, Madeline. And you already get a star on the Spartan sidewalk. Our final presentation today features Ben Orlando. Ben's academic journey began at Michigan State University, where he completed his undergraduate studies in human biology. He then pursued graduate studies at SUNY Buffalo, followed by postdoctoral research at Harvard Medical School. For the past decade, he has devoted his research efforts towards understanding the structure and function of proteins that reside in lipid bilayers. Near the end of 2020, he established his own laboratory at Michigan State University, where he now leverages cutting-edge techniques in cryo-electron microscopy to understand the structure and dynamics of membrane protein complexes in near atomic detail. His current research focuses on visualizing protein complexes in bacteria that provide these organisms with defense mechanisms against some of our most powerful antimicrobial medications. Please join me in welcoming Ben.
Okay, thank you, Dean Duxbury, for that wonderful introduction, and I'm happy to be here today to show you all about some outstanding new technology that MSU has really made some big investments in and is continuing to invest in the future that allows us to really peer down at the atomic level of some very complex molecular machines. And so today we'll be taking a gaze through the electron lens and how we visualize molecular machines such as this in 3 dimensions. So unfortunately, over the past couple of years, we've all been quite familiarized with this picture. And as much as we tend to now hate this particular virus, it's actually quite a stunning piece of artwork. And it's a piece of artwork that has some scientific inspiration. So if we were to look at the individual spike proteins that cover the surface of this virus, they have a very distinct shape and structure, right? So a structure of a protein is ultimately what is going to dictate its function. And looking at the structure of these proteins in 3 dimensions becomes critical to understanding exactly how they work. Furthermore, if we want to develop small molecule drugs that maybe target some of the proteins inside of this virus, we need atomic level insight into the structure of how these drug molecules interact with their protein target. And so this really brings up one of my favorite quotes in science from Linus Pauling, the only person to ever win 2 solo Nobel Prizes. And he said, "It is the structure that we look for whenever we try to understand anything. All science is built upon this search. We like to understand and to explain observed facts in terms of structure." And so then this really raises the question, these are minute, we're talking about looking at atoms here, right? So the question becomes, how do we see a protein structure? And to discuss that, we need to discuss a little bit some scales of biology. And so if we start off here with a typical light microscope, you can imagine looking at bacteria as they grow and divide. Eventually these will run out of nutrients and start to form spores, these white things as the bacteria burst. And the reason that we can look at this under a common light microscope, any microscope that you would find in an undergraduate lab here at MSU, is because the average bacteria has a size of about 1 micron. That's about the size of visible light that lights up this room around us and the light in a typical microscope. But if you want to go down to a much smaller level, say looking at the surface of this bacteria and an individual protein that's embedded within its lipid bilayer, we're talking 100 times smaller, down at about 0.01 micron. And if you want to peer all the way down into its atomic structure to look at individual atoms that make up this protein molecule, you need to go 100 times further, down to about 1 angstrom. And so in order to peer down at structures of this size, we really need to consider the electromagnetic spectrum, or the spectrum that makes up light. So the reason that we can look at microbes and protozoans with visible light is because that wavelength of visible light corresponds to the approximate size of the object that you're trying to visualize. And if you want to look down at atoms, you need much higher energy electromagnetic radiation. For decades, this has been done with X-rays, and we would send samples to very large particle accelerators. One of them, the closest, is probably in Chicago, and we're talking a mile diameter particle accelerator that we would then use the X-rays that are put off of that amazing instrument to probe the atomic structure of individual proteins. Over the past 10 years, there's been a monumental shift in the way in which we do this, and we've now shifted away from using X-rays to using actual electrons in a very sophisticated electron microscope. So the take-home message is visualizing protein structures requires electromagnetic radiation with much smaller wavelength than visible light. So how do we go about using an electron microscope to determine structures of proteins such as this? So in a nutshell, the way this would work is you would have a small electron microscopy grid like this. This is one of these grids held in the tip of a pair of tweezers. So as you can see, they are quite small. And coating the surface of this grid is a very thin layer of carbon that has individual holes punched into it. And we would apply our protein solution, our purified protein solution, to one of these grids, wick away all the excess solution, and rapidly plunge it into a cryogen. Typically, that is liquid ethane cooled by liquid nitrogen. And the goal is to trap your individual protein particles, represented by these black dots here, in a very thin layer of glass-like ice. This entire grid then gets loaded into a very big and very expensive electron microscope. And the sole job of this microscope, this is the one we have here at MSU, is to pass a very high intensity beam of electrons down through these individual holes. And as the electrons come down, they interact with the proteins that are suspended in this ice layer. The electrons get scattered and then recollected by a series of lenses to ultimately produce an image at a detector that looks a little bit like this. And so what you're looking at here is actually individual protein particles suspended in a frozen layer of vitreous ice. We collect thousands of these images relating to hundreds of thousands, or nowadays even millions, of these individual protein particles. We can then, from these micrographs, extract individual protein particle images in 2 dimensions. These particles can be averaged in 2 dimensions to vastly increase our signal-to-noise ratio, and then ultimately can be back projected in 3 dimensions to generate a 3-dimensional map of your protein of interest. And this technology has really expanded over the past decade. We are now routinely determining the structure of individual proteins at near atomic or even now subatomic resolution, allowing you to build atomic models of your protein of interest into these experimental density maps shown in this chicken wire here. So this technology has come of age, and how are we using it to study problems such as antimicrobial resistance, which we just heard about from Madeline? So this is the main topic of my laboratory currently, is understanding how bacteria utilize different mechanisms to fend off attack by these different antimicrobials. And if we look at the structure of a gram-positive cell wall, we have a lipid bilayer here and a very thick outer cell wall made of peptidoglycan. All of the precursors that make up this peptidoglycan layer are actually synthesized inside of the cell. They're then attached to various different lipid intermediates, flipped across to the extracellular side, and that lipid is cleaved to incorporate this small molecule, this precursor, into the growing cell wall. And this entire process needs to happen continually. If it shuts down, the bacteria dies. And so it comes as no surprise then that we have a variety of different antibiotics that target different intermediates of this overall process. Some of these you're probably quite familiar with. So if we look at bacitracin targeting UPP, this is the common ingredient in all antimicrobial creams such as Neosporin. But we also have medications such as vancomycin, which is currently one of our antibiotics of last resort, utilized to treat only some of the most difficult bacterial infections that you commonly encounter in hospital environments. And so the topic for today is going to be bacitracin. And if we look at this peptide bound to its target, UPP, what we actually find is that the peptide, shown in gray here, forms this cage around the very extracellular part of this lipid molecule, forming these interactions involving a zinc and a sodium with the diphosphate moiety on the top of the lipid species. And so the major question my lab is trying to answer is, once these peptides bind, how do bacteria sense that they are there at the cell surface and ultimately provide resistance against these peptides? And the answer is multifaceted, but the one that we're really investigating is antimicrobial peptide resistance modules, which seem to be well conserved across most gram-positive bacteria. These modules are membrane protein complexes that consist of 2 components. On the right here, we have an ABC transporter that has 10 transmembrane helices, a large, highly sequenced divergent extracellular loop, and we know that it is this component here that provides the actual resistance against the antimicrobial peptide. On the left here, we have a two-component system, which is composed of a histidine kinase that has no extracellular ligand binding domain. It does not respond to antimicrobial peptides directly, but rather is thought to sense the conformational cycling of the ABC transporter. So the way that this would work, you would have an antimicrobial peptide binding to a lipid target. This overall complex is recognized first by the ABC transporter, which then utilizes the binding and hydrolysis of ATP to change conformations and somehow mediate resistance to these peptides. That conformational cycling then signals through the histidine kinase such that it will also turn over ATP, autophosphorylating itself, which recruits a response regulator, initiating phosphotransfer, which can then allow this response regulator to act as a transcription factor, going down and binding to promoters in the DNA to regulate gene transcription. And very often what you will find in these systems is that the genes that are regulated are actually the ABC transporters that mediate resistance to the peptide. And what you get is a positive feedback loop to produce more of the transporter to deal with the need to detoxify all of these peptides. So the question is, that's all great, but we have no idea what these things look like and how they do what they are proposed to do. So to answer that question, we turn to electron microscopy. So we focused first on just the ABC transporter component, BCE A and B. We had quite good luck overexpressing these components and purifying them to homogeneity. So here's an SDS page gel showing the purified membrane spanning component and the cytosolic nucleotide binding domain. We can then freeze cryo-EM grids and take tens of thousands of individual images of these purified proteins suspended in ice, and these might just kind of look like very small black dots, but when you begin to take millions of these particles and average them in 3 dimensions, you actually get a quite high resolution view of what these purified protein complexes look like. And so we can see here an actual detergent micelle surrounding the transmembrane region of the complex, and we can simplify all of the data processing, and basically at the end of the day we get down to these experimental maps that are of sufficient resolution to allow us to build a structure of this transporter complex literally atom by atom. And so we ended up with this reconstruction here of the BCE-AB transporter from Bacillus subtilis at about 3.5 angstrom resolution. It's unlike any other ABC transporter structure that has been determined to date. And it has this highly asymmetric orientation down in the cytosolic region, and we see for the first time what that extracellular loop that is proposed to bind to the antimicrobial peptide looks like. And this is the atomic model that we were able to build into this map. If we break this down into its components, if we look at the transmembrane region here, we see 10 individual transmembrane helices, and very importantly, we identify a lipid-like density bound within this transmembrane region that places the head group of that lipid directly beneath the extracellular ligand binding domain, poising it for interaction once an antimicrobial peptide were to come in and bind. And so overall, from this, we found that BCE-AB has an unusual architecture with highly asymmetric nucleotide binding domains and a very specialized lipid binding pocket. The next big question becomes, we have BCE-AB in this nucleotide-free state. We see this lipid bound poised to interact with an antimicrobial peptide, but what happens when you add ATP? How does the transporter change conformations to actually accomplish the task that is at hand? And to answer that question, we turn to a single point mutant. So if we mutate one individual amino acid down here in the nucleotide binding domain, we can completely abolish all ATPase activity of the transporter, but we don't abolish the ability of the transporter to bind ATP. So utilizing this point mutant and going back through the cryo-EM process again, we actually now have 2D averages of the transporter bound to its fuel, ATP. And if we were to reconstruct this in 3 dimensions, we end up with another 3-dimensional cryo-EM map, now with the transporter and an alternate conformation that is induced by ATP binding down in these cytosolic domains. And so we very clearly see individual ATP molecules bound in the 2 potential ATP binding sites. And if you were to look at the overall structure, we also see that specialized lipid still bound within the plane of the lipid bilayer. This is the density showing indeed that that lipid stays bound throughout the change, the conformational change. And here we find all of these individual transmembrane helices literally clasped and collapsed around the lipid in the binding pocket. And so from these individual structures, we can then begin to stitch together literally molecular movies and watch how these machines work in 3 dimensions. We find that ATP binding induces an asymmetric to a symmetric transition that causes this pumping of the rest of the transmembrane helices and a rotation of the extracellular domain and this collapse of the helices upon one another to literally grip that lipid within the plane of the bilayer. So from cryo-EM, we've been able to stitch together these movies that allows us to then put together a molecular model of exactly how these complexes work, starting with the transporter in an asymmetric state with the lipid target of the peptide bound within the transmembrane helices. A peptide such as bacitracin would then be able to recognize the head group on the top of that bound lipid, simultaneously engage the extracellular domain, and then the binding of ATP down in the cytosol would trigger a conformational change to collapse all of those helices and hold the lipid in place, and simultaneously tilt the extracellular domain up, and we believe physically remove the peptide from its target. And then ATP hydrolysis would then reset the transporter back to its initial state. And so this is the working model we have right now for this overall complex, but it then raises the question, how do these conformational changes lead to signaling through the associated kinase to ultimately trigger gene expression? So we have some preliminary data where we've tried to overexpress all of these individual protein components and purify them directly in detergent from bacteria. We've had quite good success with this. So down here is a gel filtration chromatogram showing a shift to higher molecular weight when we co-express the kinase BCES. That shift to higher molecular weight was confirmed indeed to be due to the co-purification of the kinase module. And we could get this complex again into ice for cryo-EM imaging. And if we begin to take tens of thousands of these micrographs and millions of these particles averaged in 2 dimensions, we now get our first glimpses of what the overall complex looks like when it's complete with its ABC transporter and its associated kinase. And to put a lot of data processing down into one succinct little story, we ended up with a 3-dimensional cryo-EM map, again at about 3 angstrom resolution, showing the overall architecture for the first time of what a BCE module actually looks like. This is also the first ever structure of a full-length membrane-embedded histidine kinase, which is really quite a monumental achievement in the field of both structural biology and microbiology, and is providing us insight not only into this system, but basically across the entire spectrum of different bacteria. And so this is what the overall model of what this protein complex looks like. Quite surprising to us, what we found is that most of the interaction between the histidine kinase, BCES, and the transporter, BCEAB, is mediated almost entirely by interactions with membrane lipids. So there's actually very little direct contact between the components, and this illustrates how important the actual lipid bilayer environment is for maintaining the structure and dynamics of these highly complex machines. And so cryo-EM has some distinct advantages. It allows us from one data set to actually begin to classify out different structures and determine multiple structures from one individual data set, a distinct advantage over previous techniques such as X-ray crystallography. And so if we, from one data set, perform a principal component analysis, we actually get molecular movies that provide us insight into how the individual components of these overall complexes move in both space and time. And deconvolving all of these dynamics into function is kind of the next frontier, and where some of the new technology that's coming along is really going to allow us to dive deeper. But for the time being, we've kind of followed the same approach of biochemically trapping these complexes in different intermediates of the overall transport cycle. We now have, this is just a sampling of 3 structures, but I think we're up to 10 different intermediate states of this overall complex, which really allows us to then begin to put together more of these molecular movies and see how the individual protein components of these BCE modules move together to accomplish their overall task. And what we find is when you get this asymmetric to symmetric transition from ATP binding, that correlates at the same time with pulling over of the catalytic domains of the BCES kinase. Ultimately, we think this is somehow going to lead to autophosphorylation down here, allowing for that transition to gene regulation. And the next frontier is understanding how then bacitracin binding leads to these conformational changes that we've already observed to propagate conformational differences down throughout the entire complex. Okay. And so I'm going to stop there and say it is not me that does all of this fantastic science. It is a diverse team of Spartans made up of 2 graduate students, Natasha George and Pei-Shen Yu, as well as we have 4 different undergraduates that are doing excellent research now in cryo-EM and microbiology. I want to thank everyone at the Michigan State Cryo-EM facility, Sundar, Kristen, and Kelly. All of their input is valued on a daily basis in order to make these reconstructions a reality. Some of the data you saw today was collected over at Purdue. We also do a lot of work with mass spectrometry here at MSU, so thanks to Tony and Dan, and finally NIH for funding both this grant as well as some of the microscope time that we have utilized. And lastly, I will just finish up by saying if any of you are interested in actually seeing an electron microscopy grid or some of the massive lenses that make up these microscopes, I've brought them with me. I'm happy to show you kind of what they look like in person. So I'll end there and take any questions you might have.
Thank you, Ben. That was really fascinating. For years, the problem in structural biology, at least as far as my minimal understanding of it, was for membrane proteins was that X-ray crystallography had a problem with membrane proteins. You seem to be tossing off these structures with membrane proteins pretty readily. Is this cryo-EM just more amenable to being able to get membrane structures, or you're just really good at it? Or both?
No. It has totally revolutionized the field. So I trained as an X-ray crystallographer in my Ph.D. This technology really kind of started to take off in about 2014. Since then, there has been monumental advances both in software, hardware, and now AI is beginning to revolutionize everything even more. Crystallography of membrane proteins was a nightmare. There were many a PhD where people went with basically no data because it just was black magic in a sense. And this now allows us to do this much more rapidly. But more importantly, what I didn't show today is it allows us to do it in an actual synthetic bilayer. So now we have synthetic nanodiscs that are a small nanometer size patch of literal lipid bilayer that allows us to study the structure of membrane proteins in a near-native lipid environment. And so it has just been absolutely revolutionary from start to finish.
Thank you. I'm not sure if I didn't listen fast enough, but this is fascinating. You've demonstrated this with bacitracin. Can you give us an insight into how that would translate to other antibiotics? And my sense is that it started with the sensation internal in the cell, inside the lipid bilayer. But then when you get different antibiotics, how will that translate, or how will you perceive that? Can you pretty much turn your guns pretty readily to the various antibiotics?
That's a great question. So actually, the one thing that we are missing right now is a structure of the transporter actually bound to the antibiotic. And so that is really kind of the next holy grail for us to understand exactly how these work. And that is definitely ongoing in the lab. But the question of, can we do this with bacitracin and translate it to other antibiotics? And the question comes down to, are these found throughout the pathogenic strains of gram-positive bacteria that are plaguing our hospitals and other environments? And the answer is yes. So almost every pathogenic gram-positive has a module that looks very similar to this. And what changes is the region in that extracellular space that binds to the different peptides. And so we hope through structures bound to these different antibiotics and then computational modeling to basically build a comprehensive understanding of exactly how the different peptides would be recognized by the different modules. And then hopefully we can either chemically tailor the peptides as resistance evolves to them to beat that resistance, or potentially develop other molecules that can compete with them to allow them to do their job.
Ben, great work with the gram-positive bacteria. When you look at the gram-negative bacteria, you know, in the breakup of a cell wall, there's always been the febrile response with gram-negative bacteria. Any chance that you can figure out what's going on with that?
Yeah, great question. So that was the topic of most of my postdoctoral work. And as you mentioned, so the gram-negatives and the gram-positives have completely different cell wall architecture, double membrane system versus a single membrane with a lot of peptidoglycan, or then you come to mycolic acids even with some of the other species or strains. Totally different systems, totally different antibiotics. So most of what you saw today with the peptide-based antibiotics don't even really scratch the gram-negatives. So I think, you know, probing for which bug exactly you are targeting is going to be kind of two different avenues along this realm, and will require different medications depending on the infection that you have. Yeah.
Great presentation. Two quick points. Linus C. Pauling, Chemistry and Peace. And I don't have any fancy microbiology question, but I'm really very much interested in the chiro aspect of how this microscope works. You're talking about some significantly low temperatures. I'm sure you just don't take your ski glove and dip it in. So any insights on that would be appreciated.
Yep, so unfortunately I don't have it here, but I'm happy to show anyone later today. We can go over to the facility if you'd like to. But basically what we take is gaseous ethane, and we condense that down into a liquid form by using liquid nitrogen. And then you have a little cup of liquid ethane that the grid can be plunged into. Once those protein molecules are suspended in that ice layer, everything then takes transfer under liquid nitrogen temperatures. So
minus 170 degrees centigrade.
Yes.
Because I looked up the temperatures you were working at.
Yep.
So I assume that's all done within a hood or something like that, or you actually?
Liquid nitrogen is pretty safe, so we don't have extensive facilities. I mean, you can walk around with liquid nitrogen. If you throw it on the floor, it basically evaporates immediately. But yeah, everything is done at absolutely freezing temperatures. There is kind of this jump, so this is where the technology development is coming in, is people are actually looking at maybe doing this in liquid and potentially more at closer to room temperature to be able to follow changes in real time. That's a whole new avenue of of hardware development, but I think you'll see it within the next 10 years or so. Yeah. Okay, thank you very much.
Well, thank you, Ben. That was just an incredible finish to a wonderful day, and I think all of the speakers were terrific. So thank you to the speakers, to the Dean's Research Scholars for being here today. It's wonderful to have you at any event. Thank you, everybody, for being here. And I'd also like to say thank you to the people who put on the show, which are Corey Palmer and Sarah Ford particularly, who did a lot of the organizing, Karen Wenk, Val Ozowski, who's our communications person and does a lot of the work putting the programming and wordings together. And now it's time for you to help us out. Consider this as your homework assignment. We'll be sending you an email survey, so please take the time to let us know what it was you enjoyed most about this experience so we can continue to improve this event. For those of you attending virtually, thank you for spending your Saturday morning with us. We appreciate your support for science and MSU. Before I close, I'd like to say thank you to the people from WKAR. I really enjoyed being in this environment, and I think we should continue to work together. So with that, thank you everybody for being here. Go green!